everyone, welcome back to my channel. It's your boy Learnify back at it again with another video. I know it's been long since I uploaded last. I've just been busy enjoying uni life and finally I'm trying to upload some videos for you guys, and I'm really sorry I missed a few dates, but here we go. I'm back. Today we're going to be doing 6.1 digestive system, just learning all about the digestive system as a whole. So in terms of the digestive system, there are two parts. There's the alimentary canal and there's the accessory organs. So in terms of the digestive system, let me just draw it out. So here we have a very simplified diagram of the digestive system. The alimentary canal is basically where the food actually passes through. So food actually passes through. And the accessory organs are the organs which help with digestion but are actually not passing food along at all. So let me just draw in yellow what food would be. So food enters the mouth and it's both bitten by teeth, mechanical digestion, and also the salivary glands right here. So salivary glands, right? They release saliva, which again is used to chemically break down the food. So we have chemical and mechanical digestion. Chemical digestion is we use chemicals to break things down, break the food down. Mechanical digestion is physically breaking down. So there are three parts of the salivary glands: the parotid gland, the submandibular gland, and the sublingual gland. Next, the food enters the esophagus, which is what connects the mouth to the stomach, and there's a wave-like contraction of muscle cells by the autonomic nervous system called peristalsis, which allows food to travel through to the stomach, which is why sometimes you hear that you can eat upside down and food will still reach the stomach, although it's not recommended. Next, we have in the stomach, again, mechanical and chemical digestion. Mechanical with the rugae muscles of the stomach churning, so the stomach basically squeezes in like a bag. So squeezing in forces in, and it's able to mechanically digest food. And also you have stomach acid. We have a pH of around 2 with gastric plates releasing digestive juices, including pepsin, which is an endopeptidase, which breaks down larger chains of polypeptides into smaller chains, which can then be broken down further. Next, we have something called the small intestine, where most of the absorption of food occurs. Small intestine is known for having large surface area with villi and microvilli. So villi are small projections on the endothelial layer, and on those there are microvilli, which further increase surface area like a carpet. Right, a lot of surface area, increased absorption. Next, food reaches the large intestine, where a lot of water reabsorption occurs. So water reabsorption, which is important for not making a stool, not making the excretion very wet, which in the case of diarrhea it does, and that's because large intestine doesn't work properly, and large intestine is very important ensuring that it's maintained. Next, we also have to talk about the pancreas. So the pancreas releases digestive enzymes to the small intestine through a duct, and it is used to break down foods further. So the liver also has a role in this, where it secretes bile, which is stored in the gallbladder and then secreted into the small intestine. What the liver does is it uses nutrients to make detoxifying chemicals, which are then added to the small intestine to break things down further. Next, the food, once it reaches here, the rectum and the anus, it's excreted out. The important part of the large intestine is we prevent much water loss, which would be bad for dehydration. Next, we learn about two types of mechanical digestion. There's peristalsis, and something else called segmentation. With peristalsis, we have longitudinal muscles, longitudinal smooth muscles with this we have circular. With peristalsis, it's like a wave-like contraction, so it goes like this. So the food's here, goes in a wave, sort of. Sorry. It's like contraction in a wave, and the food's pushed along, right? Whereas segmentation, food is pushed in both directions, mixing it together properly. Okay, so peristalsis and segmentation. Segmentation is circular, peristalsis is longitudinal muscles. Peristalsis is able to move food faster, not just in the esophagus, but stomach and gut. Segmentation is a bit slower, but allows a lot of mixing, and that happens a lot in the small intestine. Next, we have chemical digestion, which is coming in three parts. So there's stomach acids, there's bile, and also there's enzymes, right? So first of all, let's go over stomach acids. The stomach has an environment of very low pH, so a lot of acid. So about pH 2. The acidic environment helps to denature a lot of proteins and break down macromolecules into smaller components. The first thing is acidic. Next, the stomach's epithelium has a membrane of mucus that protects it from the acid because you don't want the stomach to digest itself. Next, we also have the pancreas releasing enzymes like bicarbonates and also neutralizing elements. So, because it's acidic, we don't want the food to be acidic when it enters the small intestine. So, what happens is we have bicarbonate and other bases which neutralize the stomach acids before the food enters the small intestine. Next up, we have bile, which is produced by the liver and is concentrated in the gallbladder and is released into the intestines. Bile contains salts which break up fat globules into smaller droplets of fat. So fat droplets. And it's stored in the gallbladder. This is called emulsification, where the surface area is increased of fats because it increases digestion. Next up, in terms of enzymes are biological catalysts which increase the rate of reaction by decreasing the chemical energy, decreasing the energy, activation energy, and they make things work faster. So there are some enzymes here we got to mention. There's proteases and endopeptidases. So proteases is breaking proteins down, and then endopeptidases. I'm not going to write out the whole word. We have amylases, which like salivary amylase. So in the saliva, breaks down starch into maltose, and amylase here can also occur in the small intestine for the pancreatic secretions. Again, starch into maltose. Then there are nucleases as well, which break down DNAs, nucleic acids, into nucleosides. In terms of digestion of carbs, we have amylase and other chemicals. There's also an interesting fact: cellulase is present in the digestive tracts of herbivores like cows, but not in humans, which is why we can't digest grass, which is too much cellulose. Proteins are broken down mainly in the stomach, proteases, endopeptidases. Lipids broken down in small intestine with bile. Nucleic acids broken down in small intestine as well with other secretions. So here we have a cross section of the small intestine. We have the serosa, which is the outer protective layer and contains fibrous connective tissues. Next we have the muscular layer. So it contains longitudinal muscles and circular muscles, so L plus C, in order to have peristalsis and segmentation respectively. Next we have the submucosa, which contains a layer of connective tissue, so CT, connective tissue in order to separate the mucosa from the muscle layer. Then we have the mucosa, which is the highly folded end, which is where the surface epithelium reaches the lumen. This is the lumen right here, and that's where the absorption occurs. In terms of the features, here we have villi on the mucosa, and the villi, what they do is increase the surface area for absorption. And there are six main features of microvilli, and the mnemonic I learnt in school was Mr. Slim. Sorry, not microvilli, just villi in general. So the things are that the microvilli, rich blood supply, single layer to increase distance, decrease distance for absorption, lacteals, which are for sending things in the lymphatic system, so lipids, for example. Then we have intestinal glands, so exocrine pits, lots of places where juices can be secreted, and also we have membrane proteins for transport. So those are the things in the mnemonic. In terms of villi, the structure contains a lot of tight junctions to reduce occlusion. So we have an impermeable layer, so it stops substances seeping through. It essentially forms a thick layer like a sieve, so that things don't pass through around it, but rather it's filtered through and absorbed. We also have microvilli, increasing the surface area, a lot of mitochondria because we need energy to transport things actively. And we also have pinocytic vesicles, which pinocytosis means like liquid movement. This is for moving liquids and solutes from one part of the cell to the other in the small intestine. In terms of how absorption generally occurs, there's secondary active transport, facilitated diffusion, osmosis, simple diffusion. So it's what's it called? Secondary active transport is where we have transport protein where an actively transport molecule is coupled with the uptake of another molecule, so passive movement of one. So it's co-transport. And then glucose and amino acids, for example, are co-transported. In terms of facilitated diffusion, it's where energy is not used, but um, you have a passive movement of a substance by a membrane protein. Osmosis is diffusion of water across a partially permeable membrane, so you need a hydrophilic layer because water is of course hydro, and hydrophilic means likes water. Simple diffusion is where you go from one part to another, pass through. Um, then there's bulk transport and endocytosis. So bulk transport, this is where big substances pass through, not just tiny molecules like those four. Bulk transport, you have endocytosis, vesicles from the outside forming, binding to the membrane layer surrounding it, forming a vesicle inside. Bang. And then there's exocytosis, which is not really absorption, but that's another form of bulk transport. But for this one, you just need to know endocytosis. And also penocytosis to an extent, like with water absorption. So make sure you care about this bit as well. Next, we learn about starch digestion. Starch is in two forms: amylose, which is unbranched, and amylopectin, which is branched. Starch digestion happens in the salivary glands or the mouth. So the salivary glands secrete salivary amylase, which is optimal at a pH of 7, like in the mouth, and that is able to digest starch. And what it does is it breaks down amylose. Amylose Amylase breaks down amylose into maltose. So we go from a polysaccharide amylose to a disaccharide, and maltose is broken down by maltase, and maltase is. So next, amylopectin is broken down into dextrins, and that's what amylase does. So these are both the actions of amylase, and both dextrins and maltose are broken down into glucose monomers through the action of maltase. Glucose is used ATP. In terms of the pancreas, it produces a lot of amylase and puts it through the exocrine glands. It also produces hormones, insulin and glucagon, which are released from the islets of Langerhans into the blood. In terms of insulin hormone, the glucagon, they regulate the blood sugar levels. Insulin lowers blood glucose levels by increasing storage and absorption, whereas glucagon increases it by increasing release and promoting release. Cool. Now, modeling digestion. In terms of digestion, we can break down into smaller molecules. You can monitor digestion by doing various experiments. So have a Google of what dialysis tubing is. Also, yeah, that's basically it for modeling digestion. But other than that, that's pretty much it for this entire video. If you guys did enjoy, make sure you smash that like button. I try to cover all the digestive system in under 15 minutes. Digestive system topic 6.1 in IB biology, but this is pretty much what you need to know in terms of high school level for digestive system. So let's recap the main things. We learned about how mechanical digestion works, chemical digestion works. We also learned about how the small intestine functions, the stomach functions. I would say the big takeaways here are there are a lot of processes involving chemicals that you'll need to know about, and those chemical processes really do make sense because their names, for example, like endopeptidases, they literally mean breaking down large chains of polypeptides into smaller chains. It's like that. I think that overall, the digestive system is pretty important to know. If you guys did enjoy, make sure you smash that like button, leave a comment down below for video recommendations, and subscribe if you haven't already. We're so close to 200 subs. Anyways, without further ado, that's Learnify signing out.